Hi, my name is Jay Keasling. I'm from the Joint BioEnergy Institute, the University of California at Berkeley, and Lawrence Berkeley National Laboratory. And today I'm going to be talking about engineering microbes to solve global challenges. It's probably no surprise to anyone that we derive most of our fuels, chemicals, and even drugs from petroleum. We pump it up from underground, we refine it in a refinery, and then from that refinery we produce all of the fuels, chemicals, and drugs that we use on a day-to-day basis. Now, if you just look at a typical barrel of oil, roughly 80 to 85% of that barrel of oil goes to producing all of the fuel we use. In the US, that's on the order of 225 billion gallons of transportation fuels every year. The other 15 to 20% goes into producing all of the other products, from carpet fiber to paint to plastic bottles, to even some of the drugs that we ingest into our body. Now, we'd like to have an economy that's not just based on petroleum, but that's also based on renewable feedstocks, things like sugarcane, corn, and even cellulosic biomass in the form of grasses. Now, if you were a farmer and you were to grow those various crops, depending on where you are in the world, you'd send them to a biorefinery. And that biorefinery, just like a petroleum refinery, would split those molecules apart, those sugar-containing compounds apart, and transform them into fuels, into chemicals, and even into drugs. If you burned those fuels in your car, you'd still be producing CO2, like you do when you produce and burn petroleum-based fuels, but that carbon dioxide would be taken back up by the plant and incorporated into the plant containing sugars that then would be refined into fuel. And in this sense, you'd have a carbon-neutral cycle of producing and consuming carbon dioxide. Now, if we just look at the volume and the value of these molecules, at the very top of the volume and the bottom of the value are the fuels. They have the lowest value of many of the molecules we use on a regular basis, and yet they have the highest volume. Remember, I told you that's about 225 billion gallons of transportation fuel in the US alone on an annual basis. At the top of the values and at the bottom of the volume are the drugs we use. Extremely high value molecules produced in small volumes. And then in the middle are all of the other molecules and products that we use on a day-to-day basis, like carpet on the floor, the plastic bottles, paint on the wall, mouthwash, perfume, etc. Now, when we think about flexibility for substitution, we know that with our drugs, we don't want anything that's just kind of like the drug we use, but exactly like the drug that we know will cure a particular disease. But we often think that there's a great deal more substitution that we can have for things like carpet fiber, for mouthwash, and even more for fuels. However, that's not really the case. Our fuels are regulated. What we put in our tank is highly regulated because that's what gets exhausted into the atmosphere, and that ensures that our planes stay up in the altitude. The same thing is the case for plastic bottles and carpeting and paint. We need to know that those products will work the same as they've always worked, regardless of whether they're produced from petroleum or from carbon that comes from plants. Now today I'm going to be talking about engineering microbes, namely yeast and E. coli, to produce a variety of these products and to produce products that behave or are identical to the products that we would normally get from petroleum. And I'm going to start off with a story about drugs. And in this case, a story about Artemisinin, a drug that cures malaria. This guy, his name is George. I met him when I traveled to Kisumu, Kenya. Kisumu is on Lake Victoria, and it's about sea level, and it has a very high incidence rate of malaria. In fact, 80% of the population has malaria. When I visited Kisumu that day, it was very warm, but yet George came into this clinic wearing a long coat and pants, and he had a runny nose, and it was pretty clear that he wasn't feeling well. This is the clinic that he came into. It's a government-run clinic, and although it's difficult to see, there are children that are lined up outside the clinic waiting to get in. This, at the top, is a picture of the typical home, a home that George might live in. It had mud walls and a steel roof, but you can tell that the steel is not in very good shape. The nurse saw George and she knew that he probably had malaria, so she pulled out a brand new test kit that she had just had access to. She pricked George's finger, put a drop of blood at the bottom, and then followed that with a couple of drops of buffer. A few seconds later, the blood ran up the gradient. There's two lines that appeared. The top line is the control line to say that the kit worked, and the bottom line by the T is the test line, and that indicated that George had malaria. The nurse then took George, weighed him, and from his weight got a package of coartem, which contains artemisinin, an anti-malarial drug. She gave him the first tablet, and then she instructed his brother to give him a tablet 12 hours later, the third tablet 12 hours after that, the fourth tablet 12 hours after that, and so on. She repeated the instructions and sent them on their way. And then she turned to me and she said, "Jay, after the third tablet, George is going to be feeling great. And his mother is going to take the rest of those tablets and tuck them away in the drawer, because the next time he gets malaria, she may not have access to this free coartem that she was able to get. And because they're living on less than $1 per day, there's no way that she could afford the coartem or any other anti-malarial drug that would be sold in the private pharmacies." A little bit about malaria. Roughly 3 billion people on the planet at any one time are at risk of getting malaria. Roughly 250 million people are infected with the malaria causative agent called Plasmodium. And at any one time, roughly 500,000 to 1 million people die of malaria. 2/3 of them are children under the age of 5. There's a treatment, a cure for malaria. It's called Artemisinin, and it comes from this plant, Artemisia annua. And it has a great history. It goes back to 168 BC, where it was first described in the writings for being used for treating hemorrhoids. And then a few hundred years later, there are writings of it being used to treat fevers, presumably fevers due to malaria. It was largely forgotten, but in the 60s and 70s, China was fighting in Vietnam, and malaria was rampant in Vietnam, and the drugs they currently had available based on quinine were no longer effective. So Mao sent his medical corps out to find a cure for malaria. This woman here, Yu Tu, was one of the people who went out and led the medical corps. She poured through the ancient literature, and finally she found a description, shown here, for using sweet wormwood, making a tea of the wormwood, extracting the drug, and then patients drinking it. They purified the active ingredient, found out how best to extract it, and then by 1972 had the active ingredient isolated and described. In 2004, the World Health Organization recommended artemisinin-based combination therapies as the drug of choice for treating malaria. So what's the challenge? We've got this great cure for malaria. It's an ancient Chinese therapy that's now been westernized. What's the problem? Well, I alluded to a few of those challenges earlier. The first one is availability. Remember, George's mother would give him 3 tablets, he'd be feeling great, and then she'd tuck the rest away because it wouldn't be available to them. And if it were available to them, the cost would be too high. There are also challenges around quality of the drugs. There are makers of artemisinin therapies that don't put as much artemisinin in them as they should, or any at all, and try to sell them as artemisinin drugs. And finally, there's the challenge due to resistance. In a few parts of the world where they've been using artemisinin the most, resistance has, Plasmodium has become resistant to artemisinin. And this has become a big challenge. This is mainly occurring in regions around Vietnam where artemisinin was first used. Now, this is the process by which we get artemisinin. We grow plants on a plantation. We purify the artemisinin from those plants and then convert it chemically into the derivatives that are used in artemisinin combination therapies. I visited one of these plantations. I actually saw this in action. Now, the artemisinin is produced in small trichomes where it's held that are on the leaf of the plant. These trichomes store the artemisinin, and when you rub the plant, you can get the artemisinin off. When you make a tea of the plant, these trichomes burst and the artemisinin comes out. Now, the way we get artemisinin is that the seeds are planted in small greenhouses. Then once the plants grow up to be about 6 inches to a foot tall, they're taken out into the fields. They're planted by hand, where they grow up over a growing season, and they grow to be about 8 feet tall. After they've grown up, they are then harvested. The plants are chopped generally by hand. They're then laid out on tarps to dry in the sun. The artemisinin is contained on the leaves of the plants, so then they beat the plants to get the leaves off. They collect all of those leaves that are on the tarps, run them through sieves to get only the smallest leaves, and then they take it into the factory and extract it. Now, I mentioned that price and availability were a challenge. In 2004, artemisinin was chosen as the drug of choice for treating malaria. And by 2005, the price had shot way up to over $1,100 a kilogram. Farmers saw that they could make money from producing artemisinin, so they started producing lots of it. And shown in the blue bars are the amount of artemisinin produced. And by 2007, they had overproduced it, so much so that the price fell to around $200 a kilogram. Farmers then stopped planting artemisinin. There was a shortage by 2009, and the price started shooting back up. So you see these swings in price and availability. The other challenge with artemisinin is the long time it takes to produce it. You can see that by the difference in the peaks of the price and the availability. It's 2 years. It takes anywhere from 18 months to 2 years for pharmaceutical companies to decide how much artemisinin they need, to then have contracts with farmers to grow it, to get them the proper seeds that produce the most, for it to go through a growing season for the farmers to harvest it and get it back into the factory where it's extracted and then turned into the pharmaceutical companies where they then make the derivatives that are produced and used in artemisinin combination therapies. So that time window of 18 to 24 months is also a challenge. Now, what else could farmers plant if they didn't plant artemisinin? Well, they could plant many food crops, and shown here are the prices of artemisinin compared with food crops in China and Vietnam. Early in the use of artemisinin, in around 2004, many farmers in Vietnam started planting artemisinin, but those farmers went out of business as large plantations in China began to sprout up. And now most of the artemisinin is produced from plants grown in China. I mentioned that resistance is rising to artemisinin. You can see here that several countries allow the sale and production of monotherapies. Monotherapies, because there's only one drug in them, actually encourage resistance. If you have more than one drug that fights the Plasmodium with multiple modes of action, then you have a chance of containing that resistance. And as I mentioned earlier, there's been resistance documented on the Cambodia-Thailand border near Vietnam. So several years ago, our goal was to engineer a microbe to produce artemisinin from an inexpensive renewable resource that would allow us to stabilize and reduce the price, stabilize and increase the supply, reduce production times, and control resistance by controlling the supply. This was our process. We were going to replace the plant with microbes, either E. coli or yeast. Rather than producing artemisinin, we proposed producing artemisinic acid. Artemisinic acid is less toxic than artemisinin to yeast and E. coli, but there was a readily known conversion process that would allow us to convert artemisinic acid into the same derivatives that are currently produced from artemisinin. So then it could be substituted directly in the drug pipeline. This is the strategy we would use. We would engineer a microbe to produce artemisinic acid. We would then hope to get that artemisinic acid out of the microbe some way through a purification process and then chemically convert it into the derivatives. We started with E. coli as our first chassis. E. coli is an organism we know a lot about. We have a lot of genetic tools for engineering it, and so that became our first chassis. E. coli produces farnesyl pyrophosphate, a precursor to artemisinin, and so that gave us the first step in that biosynthetic pathway. Unfortunately, E. coli produces very little farnesyl pyrophosphate, so we brought in a heterologous pathway from another organism, yeast. In yeast, this pathway produces a relative of cholesterol called ergosterol that's needed for the growth of the yeast. We use this pathway to produce farnesyl pyrophosphate. It tapped into acetyl-CoA, which E. coli has plentiful amounts of. The next step in the process was to find the enzyme that converts fine or seal pyrophosphate into amorphidine, the first committed precursor in the artemisinin biosynthetic pathway. Luckily for us, that gene had been cloned, and all we thought we needed to do was get access to the gene. Unfortunately, the person who had cloned that gene wouldn't give us access, so we had to synthesize the gene directly. We also developed a number of synthetic biology tools that would allow us to increase the flux through this biosynthetic pathway. We developed scaffolds that would allow us to attach the enzymes in the biosynthetic pathway to a synthetic protein scaffold and thereby collect all the enzymes together so that they could increase the flux through the pathway. We developed regulators that could sense intermediates in the pathway like farnesyl pyrophosphate, which are toxic to organisms like E. coli if they accumulate at too high a level, and then those regulators would regulate the biosynthetic pathway. We also developed a type of debugging tool that would allow us to debug the inside of the cell and determine which genes were turned on, which genes were turned off, and what might be the cause of any toxicity or any lack of production that we witnessed. Through the use of those tools, through synthesis of genes, we were able to increase the production to about 10 million fold from where we started. And this is work that was done both at my laboratory at UC Berkeley as well as at Amyris, a company that started out of my laboratory. Now, the next steps in the biosynthetic pathway involved cytochrome P450s that would put an oxygen on that amorphidine. Unfortunately for us at the time, the genes hadn't been cloned and hadn't been described, but we knew that it was going to involve multiple steps and potentially multiple cytochrome P450s. We were worried. E. coli is not a great host for expressing these enzymes. So the first thing we did was we went about building a yeast that would be a reagent for us, an organism where we would engineer the production of amorphidine and then use it to find the P450s that might be involved in the biosynthetic pathway to convert amorphidine into artemisinic acid. We increased the expression of the enzymes in the mevalonate pathway, that same pathway that we imported into E. coli to produce farnesyl pyrophosphate. And by upregulating the genes in that biosynthetic pathway, we were able to produce sufficient farnesyl pyrophosphate and then amorphidine when we introduced the amorphidine synthase, that we could use that organism as a reagent to find the enzymes. But we still had to go into Artemisia and find the genes in that last part of the pathway. Well, as I mentioned earlier, artemisinin is produced in trichomes on the leaves. And there are basal cells that produce that artemisinin. They must be enriched in the enzymes that produce the artemisinin if that's where it's produced, and therefore they'd be enriched in the transcripts that encode those enzymes. So we went in looking for those transcripts. We developed a method to purify the trichomes off the leaf, which was a very difficult process. And then we made cDNA libraries of those trichomes and looked for RNAs that might be important for that biosynthetic pathway. But it'd be like looking for a needle in a haystack. So a very creative postdoc in my laboratory said, well, the artemisinin biosynthetic pathway, it looks a lot like biosynthetic pathways that are in lettuce and sunflower. If you look, the molecules are very similar, and because the molecules look similar, that must mean the enzymes are similar. So he then sorted through a cDNA library that we had available to us of sunflower and lettuce, looked for P450s that were unusual, and compared those to the P450s that we got in the cDNA library from Artemisia annua. And about that time, a bunch of miracles started happening. The first miracle was that the first enzyme we tried was the right enzyme. The second miracle was that rather than just catalyzing the first step in the biosynthetic pathway, it catalyzed all three steps, and the yeast produced artemisinic acid. The third miracle was really where the artemisinic acid ended up. We couldn't find it inside the cells. We couldn't find it in the broth, in the shake flask. It turns out that artemisinic acid is not as toxic as artemisinin to yeast, but toxic enough that the yeast excreted it out of the cell, turned on pumps to pump it out selectively so that it wouldn't be inside the cell and toxic to the yeast. And because the pH of the fermentation broth was relatively low, the artemisinic acid precipitated on the yeast membrane. As we increased the production of artemisinic acid, it actually comes out of solution in the fermentation broth. And this is a picture contributed by Amyris when they took their highly engineered yeast. It actually secretes the artemisinic acid outside the cell, and it precipitates in the fermentation broth. And this gives you the perfect purification. Now, the yeast that we delivered to Amyris was not ideal. It produced artemisinic acid, but not at a high level, and it grew slowly because of the toxicity. And through engineering the P450 as well as the CPR and controlling the electron transfer between the CPR and the P450, Amyris was able to increase the production and give us a host that's highly productive. That host is now being used by Sanofi, producing artemisinic acid in large scale in Eastern Europe. That artemisinic acid is then converted into artemisinin using a light catalyzed process, much like we think occurs in the plant, where light catalyzes the conversion of artemisinic acid to artemisinin. And this is the full-scale facility that Sanofi built in Gariso, Italy, where they're producing the artemisinin. That artemisinin, shown here, pallets of it, is then shipped to Morocco, where it's tableted. And there is the final product right down there. 3 million treatments had been delivered to Africa as of about January. By May of this year, 16 million treatments had been shipped to Africa. And Sanofi will have the capacity to produce between 100 to 150 million treatments on an annual basis. That's roughly 1/2 of the world's needs. Now, what I've told you is a story about producing artemisinin in yeast and E. coli. There are many other products that we might want to produce, things like fuels. Now, as I mentioned earlier, if you produce fuels using engineered organisms, and those fuels are derived from renewable resources like sugars from biomass, the carbon that you put into the atmosphere is carbon that the plants took up in producing that biomass. So you have this possibility of having carbon neutral fuels. Well, we had an organism engineered that could produce hydrocarbons. Now, ethanol is one of the fuels that's been most widely used and is commercially produced in the US and supplemented into gasoline, but it can't substitute for diesel and jet fuel. We need a fuel that behaves exactly like the fuels we get from petroleum. Well, we had in our hands a host that would produce hydrocarbons, and we thought we could just convert that into a fuel-producing host. Here are some of the fuels that could possibly be produced using an engineered organism. They include short-chain hydrocarbons that you'd find in gasolines, long-chain hydrocarbons that you find in diesels and jet fuels, and even aromatics that you find in all fuels. So we went about engineering that yeast and E. coli that we had first engineered for producing artemisinin to now produce hydrocarbons that would have similar properties to the hydrocarbons we get from petroleum. And that engineering involved replacing the genes that were specific for producing artemisinin with genes that are specific for producing the hydrocarbons that we would use as fuels. Now, one great thing about these fuels, which is very similar to the story I told you about artemisinin, is that they are pumped or leak out of the cells. And shown here is a tube that had E. coli growing in it, and you can see at the top there's an oil film at the top of that tube. And then shown in this picture are E. coli swimming in their broth, and you can see the blebs of fuel that are being excreted and are collecting. Now, this makes the purification process for these fuels really simple. You grow the cells up on the sugar, they produce the fuel, excrete it, and it floats to the top. It's a lot like making a vinaigrette. The oil floats to the top, you skim it off the top, and if you've engineered the biology right, you can put that into your tank and drive away. Now, I've talked to you about fuels and drugs. I want to just mention the possibility of engineering microbes for producing all of the other chemicals that we now get from petroleum, things like mouthwash, perfumes, carpet, and paint. As I mentioned earlier, about 15% of a barrel of oil goes into producing all of those products. And yet that 15% of the barrel of the oil generates as much profit for companies as the other 85% because of the higher value of those commodity and specialty chemicals. But again, we have to produce products that behave exactly like the products that we get from petroleum, not things that are kind of like it. But shown here are just a variety of the products that we currently get from petroleum. And this is a pared down list. The list is vast and extensive. But we can use a lot of the same synthetic biology that I talked about earlier, engineering the microbe to produce identical precursors to the ones you get from petroleum that could then be incorporated into all of these products. And there we'd have a renewable economy. Now, one of the challenges that we have in engineering biology to produce all the products we want is that it's expensive and time-consuming. That project I described about engineering yeast to produce artemisinic acid, that took $25 million just for the research and development alone, and about 150 person-years of work to get it accomplished. We need to be able to do these projects with a 10th or a 100th of that if it's ever to compete with petroleum processes. Why did it take so long? Well, just to engineer that yeast to produce artemisinic acid, we needed over 40 components, the genes, all the regulatory switches. We had no standards for those connections, no accurate models, and no details could be ignored. And what's more, there was no bioshack to go out to buy any of those components. Biological engineering is slow because we design something, we then go into the laboratory, we build it, and we test it, and hopefully we learn from our mistakes. But one turn of this cycle can take weeks, if not months, and many years ago it took a year or more to go around this cycle. We need to turn the crank faster and in higher throughput so that eventually we can design once and have it be correct after the first design. What's more, companies don't share when they've gotten good at this process. Amyris became very good at engineering artemisinin. DuPont became very good at engineering 1,3-propanediol, but when they become good at that, that's a trade secret, and they keep it in-house and don't share it. So the timelines don't get shorter and shorter as we would hope they could. We really need the biotech industry, the next generation of biotech industry, to look more like the microelectronics industry so that we can have really powerful things like we now have computers and cell phones that won't cost as much to develop as they currently do with biology. And so I'm advocating for a next generation of biological foundry. Just like we have silicon foundries, we need to have biological foundries that will allow any user to come in, design on the computer what they want, send it off to a foundry to be built, and come back to them and work as planned. This is my goal and something that I think the future holds. I think there's a lot of promise in this, and it's something we have a head start on. I'd like to acknowledge all the funding and the work. There was a lot of work from people in my laboratory, Amyris and Sanofi, for the development of the semi-synthetic artemisinin, the work at JBIE and in Sinbert for the fuels and chemicals, and I'd like to thank all the funding from the Bill and Melinda Gates Foundation, the Department of Energy, and the National Science Foundation for all the work that this enabled. And I'd like to thank you for listening to this video.